So all of the techniques that I'm going to talk about today have in common that they all involve antibodies. And so they all tie into what we're talking about in class because of the fact that they involve antibodies. Some of them are things that we think about as sort of immunology. Some of them are techniques that you might think of as, oh no, that's not an immunology thing, that's something they use in other fields. But it involves antibodies, and a lot of the concepts are actually the same in terms of how these assays work. So that's kind of what's tying all of this together today. This is a table from the textbook that actually talks about some of the assays that are out there that involve antibodies. What you should notice is that there's a lot of them, and they have some variation. One of the places where they vary is in their sensitivity, which means how much antibody or antigen can be detected. Do you need to have micrograms of antibody? Do you need to have nanograms of antibody? Do you need to have picograms of an antibody? Same ranges for antigen. So there's a big range with these assays in terms of how much antigen or antibody you need to get the tests to work. The different tests that we're going to talk about also vary in that some of them are testing for antigen, using antibodies as like the fish hook to find the antigen. Others are testing for antibodies, using antigen as the fish hook. And so you can use either side of this relationship. You can use either your antigen or your antibody to help you find the other. So you could do one of these assays to find antigen. You could do one of these assays to find antibody. The other thing that really varies here is what kind of sample you have. Is it like a liquid? Is it like a solid, like a piece of tissue? You know, what exactly is this material in terms of your sample? So those are kind of the places where some of these assays are going to vary. So the really most simple of all of these assays using antibodies are assays that are called either the precipitation or agglutination reactions. And they're all pretty much the same. I'm just going to lump them together for our purposes. What you can notice is that a lot of these assays are the ones that require a pretty large amount of antigen and antibody in order to make them work. And this is really the basic idea between the precipitation or agglutination reaction. So the idea of a precipitation and agglutination reaction is that if you have antibodies and antigens mixed together at a concentration that is just right, they make a big clump. You can see that you've got this antigen bound to two antibodies. This antibody is actually binding to two antigens, and this one's also got two antibodies on it. So you get this sort of big higher order structure, and that can make a clump. If you have no antibodies or few antibodies, then you don't really get the clumping. This would also be true if you had 0 antibodies. If we have very few antigens or no antigens, you don't get the clumping. But if you have just the right combo of these, you get a clump. So you don't have to do anything special to your antigens or your antibodies. You literally put them together and look and see if you get clumpy stuff. This probably sounds very old-fashioned and not useful, but this is done every day in the clinic. This is actually a very frequently done assay. And the place where it is done really frequently is dealing with blood group antigens. So all of your red blood cells have some carbohydrates on their surface. Those carbohydrates are shown here. So basically there's this carbohydrate string. And there can be nothing at the end of it or there can be something at the end of it. So it's basically about this terminal sugar, or its lack thereof. If there's no terminal sugar, it's called O. If there's this kind of terminal sugar, a GalNAc, we call it A. If there's a galactose, we call it B. That's the difference between A, B, and O blood groups. And so really, you can think about the blood cells in individuals with these different blood types, as shown here, in terms of antigens. The person with the O doesn't have this particular sugar antigen. It doesn't have a terminal part of a sugar. Yes, it has plenty of other antigens, but the antigen we care about right now, it doesn't have. So no antigen. A person with type A blood has just the A antigen. A person with type B blood has just the B antigen. And a person with type AB blood has both the A and the B antigens. Do you want to hear the worst joke I've ever heard? Yes. Although it may slightly be easier, I read it, I first saw it like online and saw it written. It might make more sense written, but I'll just tell it to you anyway. And the person who was telling it to us said that it was the nerdiest joke that ever existed. So, a priest, an imam, and a rabbit walk into a bar. Make sure you think about what I just said. The priest, the imam, and the rabbit walk into a blood bank. That's right, they walk into a blood bank. The priest says, I think I'm a type A. The imam says, I think I'm a type B. You already got it. The rabbit says, I think I'm a type O.
I think it's better on text.
It's a little better on text. Because a rabbit is not like a priest and an imam, but a rabbi is. And so it's a typo. It ties in with what I'm saying here. See? Thank you. So, this is sort of the general idea of differences in blood group antigens, and we can actually just blood test people really quickly with a quick precipitation reaction. Basically what we're doing here is we're looking to see if people's blood cells have these antigens on their surface. So the way that this works, I am not going to let you guys blood type yourselves in class because there are a number of biological safety as well as other issues in so doing. But basically what you do is you have a card and that you drop some antibodies on the card. So you use a little dropper bottle and you put antibodies on the card. You put multiple antibodies. I guess I have to draw multiple to make this work. So again, this is like a piece of paper, and I just put like a solution with a dropper bottle that contained antibodies on it. Then I put a drop of my blood on each of the 4 dots. This is an antibody against the A antigen. This is an antibody against the B antigen. This is an antibody against the Rh antigen, which is whether you have like positive or negative. So it's a different antigen. And this is a positive control. And you put a drop of your blood on each circle. And then the blood either spreads out across the whole circle or it clumps. If it spreads out across the whole circle, it's basically not binding to any antibodies. if it binds to the antibodies, it makes clumps, which look like little dots. And actually, sorry, this is not a positive control. This is a negative control. Good job, Sulf. So this is no clumping on the end. So this one's not going to clump no matter what. You're always going to have the blood spread out. And so this lets you use antibodies to look for the presence of antigen. Specifically, we're looking for the presence of those antigens on the surface of red blood cells. So this is a version of this. So this is actually when I blood typed myself using a quick blood typing kit. I would say, basically, you take your blood, you drop it on the card, and you leave it there for about 5 minutes. And so if you're in the hospital, you need blood, they can just do that right away to know whether they need to transfuse you. Say if you're in a car accident, you can't talk to them about what blood type you have. They just do this. In 5 minutes, they know what blood type you have. Then they can go ahead and do things easily. So this is, like I said, this is done every day. The fact that it's a super quick and easy test is really easy. So based on these data, what blood type do I have? Yes, Hannah. B positive. So my blood did not clump with the A antibodies. My blood did clump with the B antibodies. My blood did clump with the positive, the Rh positive. My blood did not clump with the negative control. And so that told me that I had B positive. Just as a further example, the NobelPrize.org has a blood typing game on their website where you can figure out details of blood typing. I'm not going to go through all of the details of this whole game, but this is really what happens. We have our patient who needs a transfusion. We need to take some blood. So we're going to hope that we can take blood from our patient. There we go. All right, I got blood from my patient. Now I'm going to take the blood and put it with the 3 different kinds of antibodies. So here, this is on a test tube, not on a card, but it's the same idea. So you can see the data here. You can see what happened in the test tubes. So what blood type is this patient? Rishi? AB negative, because we got clumping with the A antigen. We got clumping with the B antigen. We didn't get clumping with the Rh antigen. And so these tubes originally contained antibody. We're getting the clumping. So AB negative. And we're right. It then will go into another section about transfusing this patient, but we're not going to deal with that right now. But this is actually what happens if you are in that situation. And it's all based on this very simple antibody-antigen interaction. So this information about using these antibodies and antigens is actually pretty useful. Then we can kind of move into another group of techniques. So the next group of techniques are like fancy. They're not very fancy. But the thing that they all have in common is that you generally have to modify your antibody a bit. In some way, your antibody has to be modified. And basically, the way that the antibody is modified is that you stuck something on the Fc portion. So basically, you just stuck something on the end. And so for all of the techniques we're going to see, now we had to modify the antibody. Before, we really just used plain old antibody and antigen and looked for the clumping. The most sort of simple, straightforward way you could think about doing this is with immunostaining, as is shown here. I think that Dr. Miller did this in some 250 years, but not all of them. So some of you may have seen this, some of you may not have. But the basic idea is that you put some indicator on the FC portion of your antibody, and you use that to help you find antigen. So here we could see, oh, I've got this patient sample. I want to know if it has the virus in it. So I'm going to bind my antibody. My antibody has a fluorescent tag, so I can see if the antibody is present or not. There is a washing step, so if it didn't stick, if we didn't have good antigen-antibody interaction, it gets washed away. But if the antigen was there, it sticks, doesn't get washed away. And so then we can actually look at something like this, where we can see, oh, look, in this tissue section, there are lots of cells that are positive for antigen. This is really useful in detecting antigens from different types of systems, although generally you are looking at something on a slide, and you're looking under a microscope. It's great because it can say, is my sample positive, negative? It's great because it can say, like, what part of the cell, or what types of my patient cells, or which structures. But you got to have a microscope. You got to have a sample that can go under a microscope, all that stuff. So pros and cons. The other kind of very famously useful technique that comes up here is a technique called a Western blot. Again, you probably have seen a Western blot with Dr. Miller. It's really the same thing as immunostaining. It's just that you don't start with stuff on a slide. You start with stuff in a gel. So what we do is we can take a sample of cells where we've lysed the cells, we've got all of the antigens out. Usually here we're talking about protein antigens, and we can run those on a gel. They'll be separated by size. The gel can be hard to work with, so we move those separated proteins onto a piece of paper, so they stay separated. And the paper is easier to work with than the gel, which rips if you look at it funny. And then you just put enzymes on the paper. If the antigen of interest is there, the antibodies bind and you get a band. If the antigen of interest is not there, the antibodies don't bind and you don't get a band. And your antibody generally has something on its FC portion so that you can see those bands. Because I don't know about you, I can't just see antibodies on a piece of paper. You actually need to be able to visualize them in some way. So this is all a Western blot. As you can see, it's actually the same technique as immunostaining. It's just you did a gel first. So what can you imagine as being sort of pros and cons here? Yeah, Jamie.
There's more specificity as to what you're recognizing with the antibodies.
Why is that?
Because the proteins are distributed by size, you can say like, oh, this protein that I'm identifying is around the same size as these proteins, so it could be this.
Yeah, so you get some size info about the protein you're looking at, which you didn't get in the other one. You also don't have to have a sample that's on a slide. You can have lysed cells that were on slides. What's the con of this one? Yeah. Equipment. So this is, this one takes a couple days. It's not one you can really easily fit into a lab period section. What other cons can you see with this? Yeah, Sydney.
And there's three, if you talk about one that's an indirect target for the antibodies to link up with, if you denature the protein, then since it has to do with the 3D structure of the antigen, you won't get antibody binding.
Absolutely. So if we are denaturing our proteins, we could destroy our epitope. And so we need to have a fair amount of knowledge here to make sure that we're not destroying our epitopes. We also don't get great info about where in the cell is the protein, which we did in the other one. So you have to make a choice of which one am I going to use, which one's going to be better for my purposes. You actually can use Westerns for both antigens or antibodies. So this Western is showing you where you are basically saying, is yellow protein present? You're looking for the antigen. There are ways where we can take known antigens, run them out, and then take serum from a patient and say, does my patient have antibodies against this antigen? So you can sort of flip it that way, and you can use your Western to detect antibodies as well. That's actually how HIV testing is done, is that they're looking to see if you have antibodies against the HIV antigens, which they've run out on a gel. So this one could go to test antigens. This could go to test antibodies, depending on what your exact experimental question is. So this seems to make sense, right? All right. So there are other modifications of this same theme as well. The next modification that I want to tell you about is perhaps the trickiest part of this whole day today. It is the thing that I am most worried about out of everything that we are doing today. Basically what, some of you guys have done, you guys have all done Westerns with Dr. Miller. If you remember, she had to split that Western up over 2 lab periods. In general, when you do a Western, it takes a couple days. If your goal is to test a patient for antigen or antibody, 2 days might be too long to wait. There are lots of situations where you need an answer right away. So people have come up with modifications of this that can be done very rapidly. There are different types of rapid tests. And I'm going to talk a little bit about some of those rapid tests. Particularly, the one that I'm going to talk the most about is called a lateral flow assay. And I'm going to do a demonstration of a lateral flow assay for you, which is the one that, like I said, this is the thing that I am the most worried about today in total. You have a little packet in front of you. That packet is the packet for something called the Binax Now COVID antigen test, which is in fact a version of a lateral flow assay. This is in fact you can buy these at Walmart or at Walgreens or whatever thing. It's 2 tests for $25. I feel very strongly that is way too expensive, but anyway, that's how it is. And this box has antibodies in it, and involves a test using antibodies. So I'm going to test myself and sort of walk through the whole process for you. I have no reason to think that I am going to be positive. With my luck, I'm now going to test positive, because that's how things have been going for me lately. So that's why I'm really nervous about this one. So the first steps that I'm going to do are on this page that you see on the 3rd page. I'm going to sort of walk through them quickly so that you can see me do them. But what you will notice is that part of it involves a nasal sample, which I am going to go do in the hallway. So I do not have to take my mask off and stick things up my nose in front of the class. While I am taking my nasal sample, you should go ahead and read through this. And in particular, the thing that I want you to take a look at, in fact, it's actually more important. I mean, you go ahead and read through it, and we'll talk through the details of what are happening. If you look at actually the protocol part, so if you flip through the protocol while I'm doing my nasal sample, you'll see there are a couple places where there's like, at the bottom, it says note. And it tells you something you could do wrong for a false positive or a false negative. And so as we talk through how the assay works, I want to hit on why those things would give you a false positive or a false negative, like how those could mess you up. So take a look at those, kind of see what those notes are. There's some notes, for example, on steps 3, steps 5, and steps 9. The one on step 9 is actually pretty important. So make sure that you take a look at those. And I'm going to show you the first couple of steps, and then I'm going to go swab my nose. So in the Binax test, you get this little card thing. So you get basically this little card. You can see it has a window here, which is where the results are going to be. You also have, it also opens up. You can see, if you look closely at the sort of back of the window, there's like a little skinny piece of paper. So you can see that little skinny piece of paper. That paper is going to become important. We're going to be, in fact, that paper is the nitrocellulose membrane that's drawn up there. You also can see that there are two little dots here. Okay, and so those two little dots are also going to become sort of important. And so the first step of this protocol, you will also note it says, if possible, wear gloves. Perk of being a lab person. So I removed the test from and laid it flat on the table. I will now open it wide on the table. And I'm going to use my little dropper bottle that has some liquid on it. And now it wants me to put my dropper bottle over the bottom hole and put 6 drops of liquid onto the hole. So I have done my dropper bottle. I now need to take my swab and go swab my nose. I shall return. Okay. I swabbed both nostrils. It says check, did you swab both nostrils? Yes. Now I'm going to take my swab tip, and I am actually going to put it into the bottom of this bottom hole. It's going to go into the top.
Very nice.
And I am going to turn 3 times. 1, 2, 3. Now I'm going to stick this together and press firmly and let it sit on the table. And then I'm going to wait for 15 minutes. During the 15 minutes, I'm going to tell you about the science behind what's going on in there. I'm also going to set a timer for 15 minutes. Okay. All right. So now let's talk about exactly what's going on in there and how this is going to help us. So that little piece of paper that I showed you, that nitrocellulose membrane, has a few regions on it. Part of it over here has absolutely nothing. And really, it's a piece of paper. It's a piece of membrane like you would use in Western or somewhere else. It's really, you can imagine it as being pretty much like paper. But there are 2 areas at the edge that have stuff on them. One of them is right now in my little window, says test line next to it. The other says control line. And in the test line, we have antibodies that binds to SARS-CoV-2, that binds to this virus. We also have a chemical that could change colors if it is in the presence of another chemical for a little while, okay? So there's also kind of this chemical here. It's invisible. On the control line, we also have some antibodies. These antibodies bind to other antibodies. They're antibodies against antibodies. Specifically, they bind to a type called IgG. And we also actually have the invisible stuff here. Finally, we have one other important region on this test, which is a region up here. This also contains antibodies. That are against SARS-CoV-2. And specifically, they are of the IgG isotype. So these are specifically some IgGs against SARS-CoV-2. And these are modified on their Fc portion to have the thing that converts this chemical to make a color. If it stays for a long time, it takes a little while for that reaction to occur. Chemical that converts that other stuff, the scribbly stuff. Okay? So this is what that test was like before I started using it. If we just had a piece of paper and had some antibodies on them, nothing would happen. It would just live happily ever after. But if we put some liquid on this, then substances can sort of move around here. Particularly, they can move this way because of the way the paper is set up. So we put some liquid here. And then we put our sample that may or may not contain the SARS-CoV-2 antigen there. So remember I put the little drops on? That was me putting on some liquid. And then I put the thing on my nose. And that was the sample that may or may not have contained antigen. And then the liquid started to flow through this paper. And the first thing that happened is the liquid hit this region. And it picked up all this stuff. So all this stuff started to flow this way too. It's a lateral flow test. So let's imagine first, what happens if I have SARS-CoV-2 virus? Just in this piece, right, what's going on here? Yeah, Jamie. Yeah, so these antibodies are going to bind to that antigen, right? All right, and then we're going to keep flowing. What's going to happen when all of these materials get here? to this little stripe. Yeah, Jamie.
The antibodies will also bind, and there's going to be no.
So now these antibodies are also going to bind to the virus. This antibody and this antibody are going to be held in close proximity to each other because they have a virus in the middle. It's like sandwiched in the middle of them. So the virus is going to hold these antibodies together. And that's going to allow for a nice sustained color change. And as a result, this test line is going to suddenly go from invisible to pink. Does that make sense? Okay, things are still going to keep flowing. So what's going to happen as things keep flowing? Yes, Jamie.
Control is going to, so that antibodies against IgG are going to bind, and then the control is going to.
Yeah, so we're going to still have some of this mixture binding. There's going to be still some more of these antibodies that didn't all get trapped here. And they're going to come over here, and they're going to bind to this antibody. This antibody is going to catch them now. And this antibody will now bind to this one. We've got our chemical. We're going to get this one to change colors. And so that's the positive control that says this whole flowing business and antibodies binding and chemicals changing worked. So we get this line that says the test worked. We get this line that says positive or negative. If my sample, we hope, has no SARS-CoV-2 in it, it's still going to flow here. The liquid's still going to flow. It's still going to pick up these antibodies and everything in this area, and it's going to keep going. When it gets to this line, there's nothing to get caught. There's no virus, so nothing's getting caught on these. The chemicals aren't going to stay there very long to give you the really nice clear line for color change. Everything's going to keep flowing. Those antibodies are going to get caught here. And in fact, we are still going to get a positive line that tells us that this worked. And this is all shown on this slide here as well. The same basic idea of what's going on. So, it has not yet been 15 minutes. Should I look at that card? No. Why not? Yeah, Justin. Okay, so it might still be going down. We may not have had flow all the way to the end yet. There's actually another kind of piece of that, but it's all related to what Justin's saying. Yeah, Addie. So I don't know the test is accurate until everything has reached the control line. Yeah, Jamie.
With that, like more specifically, if you haven't reached the test line yet, you can get a false negative.
Mm-hmm.
And if you have gotten past the test line, but it hasn't been very long, you can get a false positive from that same.
Yeah, so in fact, if you look at these tests, and I have done a bunch of these tests over the past few months, I can tell you this is really what happens. They all come back positive at like 1 minute. And that's because the stuff has flowed. You're getting like a temporary reaction, but the antibodies haven't been held there very long. And they flow away as time goes on. So right at the beginning, it's actually like positive here. It's all like pink right here. If you look right at the beginning, because you don't have everything flowing into the proper places yet. And so you want to wait until you've had time for the flow to happen completely through this process. You want to wait until you've had time for the color change to happen appropriately, all of that. And so that's kind of the general idea of how this works. And so again, you can see that these rapid tests are all based on antibodies and all of the antibody-antigen interactions that we've seen so far. In addition, just so you know, this is also how a pregnancy test works. It's a little piece of paper in the pregnancy test, and you have urine on it which is a liquid, that if the woman is pregnant, she has a specific hormone, which is the antigen. So you're either putting liquid with antigen or liquid without antigen on this. It flows by antibodies, and it's the exact same thing. Sometimes they're fancy, and they make it so instead of the two lines being parallel, they cross, so you get a plus or a minus. But it's the same thing. And so this idea is used for rapid testing for antibodies, and or for antigens. There are some modifications for antibodies all the time. Obviously, with a pregnancy test, people have been getting pregnant slightly longer than they've been getting SARS-CoV-2. And so we've had more time to kind of optimize and make it quicker and make it cleaner and make it not have quite such a long set of instructions and like, if you do this wrong, you're going to ruin everything. And if you do this wrong, you're going to ruin everything in the way that some of this SARS-CoV-2 book writes things out. But it's the exact same principle. All of these are just precipitin reactions where you're looking for the precipitation of that material when the antigen and antibody come together. Yeah, Jamie?
Are there concerns with these types of tests, like the antibodies not reacting over time?
So Jamie wants to know if there are concerns with antibodies not reacting over time. And so, yes, absolutely. And so how do you think we deal with that? Yeah, all these things have expiration dates on them. So the ones I have expiration dates of early December. And so yes, these are all going to expire because they are using this biologic protein that can expire. Why? So one other thing that they have in this little booklet is, I guess you can't see it as well on the photocopy. Karen, question for you. I want you to look at this little thing. You can see in color easier here than on the thing, where you are checking your result. So this will be on the 3rd page that you guys have, I think. No, the 4th page that you guys have. In the top, interpret results. So when you look at that, what does it tell you? What does it say, Karen?
So it says if you see 2 pink purple lines, one on the top half and one on the bottom half, this means COVID-19 is detected.
Okay, then what does it say under that?
Look very closely. The bottom line can be very faint. Any pink purple line visible here is a positive result. And there's different ways that it can look.
Okay, so then they show you some examples of things that are still positive results. So why is there that variation? Yeah. So it's how much antigen did you have? Do you have a lot of virus or a little virus? That's your antigen. Also, does your virus bind very well to the antibody that this company used? Does it bind poorly? And so, if you have a slightly different version of the virus than other people, it might bind a little bit less well. And yeah, one thing that always kind of freaks me out a little bit when I read this, just is because when I'm reading all of the things that can go wrong, it talks about how if you touch certain parts with your fingers, you can get a false positive. And so I'm kind of, there are a couple that I'm like, really? You sure about that? So I would love to get some more info from this company on their antibodies that they're using and their reactivity. Because I'm like, why should my finger also make it positive? But I use gloves, so it doesn't matter. All right, ready, Eileen?
Ready.
Oh. They do. They totally look like lollipops.
Oh, they do.
I'm good. So, just so you guys know, this is kind of what the end result looks like. Hooray, I lived. So another really important technique that's, again, a very similar use of the same kind of ideas is known as an ELISA. An ELISA is known as an enzyme-linked immunosorbent assay. There are 2 general different types of ELISAs that exist. One of them is shown here. The other one is shown on the next slide. And so officially, The ELISA that is shown here is called a sandwich ELISA. The ELISA that you will see on the next slide is called an indirect ELISA. In a sandwich ELISA, you are looking to see if antigen is present. In an indirect ELISA, you're looking to see whether antibody is present. And so with your sandwich ELISA, you first will take a little plate here, and you will put some antibodies on it. So you'll coat it with antibody. You can then take your sample that may or may not have antigen and put that sample on. If the sample has antigen, it will bind. If the sample does not have antigen, it will not bind. Then you wash off anything that is potentially remaining. So basically the antigen and antibody will stay. Anything that's unbound will go away. You can then add another antibody. The reason why we call this a sandwich ELISA is because the antigen gets sandwiched in between the antibodies. You're making an antigen sandwich with the antibodies as the bread. You can see that this antibody has something on its FC portion. You can then add a chemical to get a color change. And this can be sort of quantitative in terms of how much antigen there is will actually influence the amount of color change, because it will influence how much antibody is bound here. And it will look just like this. And really, it's this same principle you just saw on a piece of paper, now in a little well of a dish. This one takes longer than the lateral flow, but it can be quantitative. So you can actually get amounts on this one. You can't really get an amount on here. So you can make a standard curve and get amounts with an ELISA. Again, you're going to probably be using kind of a liquid sample here, just like you used a liquid sample on your lateral flow. And so ELISAs are pretty much like the workhorse of immunology. I have definitely done a lot of ELISAs in my life. I've had students in the past ask me whether I would buy them an ELISA robot because they were tired of doing ELISAs. And I told them that during my 4 years as an undergrad, largely what I did was take HIV vaccine candidates, inject them into mice, get antibodies from mice, and then do ELISAs on them. And as far as I understood it, as those 4 years when I was an undergrad, I was an ELISA robot. So, I mean, I don't even know that you can buy one other than, like we're ELISA robots. What are you talking about? Anyway, so this is one type of ELISA. Again, this is the sandwich ELISA where you're looking for antigen. Alternatively, we can do something called the indirect ELISA. And in the indirect ELISA, you're looking for antibodies. So here you actually put known antigen into the well. And then you add serum from your patient, which may or may not contain the antibody of interest. You wash, and then we add another antibody that binds to the FC portion. It's really just like this anti-IgG thing that's up here. This one, again, has the enzyme on it, and we can get the same kind of color change. And we usually refer to this antibody as the secondary. It was a secondary here, too. All right, so what I'm eventually going to have you do is you are going to do an ELISA. And the ELISA that you are going to do is going to be doing some testing for SARS-CoV-2. So it's going to be a separate SARS-CoV-2 testing ELISA. However, before I have you do that, I'm going to finish the slides. I've been going back and forth. I'm like, should I break and then come back, and break and come back? I think it's just easier to do the whole thing and then have you guys do the ELISA. So that's great. And all of the rest of this is really going to be largely about one particular technique for the rest of the time I talk now. And this technique is related to all the techniques we've just talked about. And so part of the reason why I set this up this way is because this technique freaks people out. They're like, oh, it's so hard. It's not. It's actually the same concepts you just saw, just applied in a slightly different way. So if you get everything we've been doing thus far, you can get this, no worries. And this technique is used to help us solve a problem that you have already been exposed to, which is that sometimes it can be hard to tell the difference between leukocytes simply by morphology under a microscope. So if you think about when you distinguish leukocytes by morphology, did you feel like you were 100% sure at all times on all those cells? No, it sometimes could be pretty hard, right? What do you think was one of the ways that you could have improved your data that you had? So if you think about the data you had, you made those little tables. What could have improved those data? Yeah, Sydney. Okay, so maybe being able to see or get more information about the cells? Sure. What else? Yeah. Combining multiple sets of data, which is a sort of other way of saying get more data, count more cells. So if you had counted 1,000 instead of 100, you would have had more data. So if you had more information, either about the cells or maybe you'd been trained more, or if you counted more cells, you could have gotten better data, right? So how would you have liked it if I told you in that lab to count 1,000 cells? But how would you have felt if I said, your job is to count 1,000 white blood cells? Sad. I'm getting lots of variations on sad. Why? It's time consuming. It's a lot, right? Those cells are really rare in the blood in the first place. And so if you want to actually start to understand what's really going on with those cells, you need to look at a lot of sample. You need to look at a lot of cells. When I did experiments like this in graduate school, I would generally look at I would say absolute minimum would be 10,000 up to 1 million cells per sample. So I want you to think about how much fun you would have counting 10,000 to 1 million white blood cells in those samples. Excellent facial expressions about how much you would really not like to do that. There's one other piece of info that we need to think about a little bit. If you recall, you can think about the different types of cells that you looked at in that lab. How many B cells, which are the cells that are producing antibodies, did you count? Yeah, you didn't show me about any B cells, because B cells are a type of lymphocyte. So are T cells. In fact, there's a ton of types of lymphocytes. So if we wanted to make a call about how good is your T cell response after getting vaccinated, you couldn't have done it in that experiment. There are tons of types of cells. So this is actually an underestimate of the different types of lymphocytes there are. And again, this is just a pie chart, also kind of estimating types of lymphocytes. And what you can see, the moral of the story is there's a lot of them. And none of that was possible with what you did in that lab. And so there's information that you can get from that, but there's also information you can't get. So when I was in graduate school, I did some experiments where I infected mice with some viruses or gave them some vaccines and tried to make graphs like this. In fact, I have published graphs that look like this. It's just a cartoon version. So what you can see is that I gave my mice vaccines at a couple different time points, and I specifically wanted to measure the size of their immune response, and I cared about how many virus-specific CD8 positive T cells they had. So I wanted to make a graph like this, where at day 0, they had this many, and at day 4, they had this many, and at day 7, they had this many, and day 10, they had this many. And I wanted to make a graph like this. Said graph exists. It actually goes out for like a few hundred days. But what that means, for me to make that graph, I had to have a way to tell the difference between the CD8 positive T cell that responded to my virus of interest, HIV, and tell the difference between that CD8 positive T cell and the one that bound to flu. And I needed to know how to count those T cells when there's also CD4 positive T cells and neutrophils and NK cells and gamma delta T cells and B cells and monocytes and all of those things. And way back in the day when I made this, I even made this with like the right proportions of all of those cell types. And now I look at that and I'm like, oh, that was really cute of me. Like, wouldn't that be an interesting thing to have time to do now? So I had to be able to just count these and make a graph out of this mess. And if you think about the lab that you did, that was not an option for how you did that. So the way that we do this is because we know that leukocytes have different proteins on their surface. Immunologists have defined different leukocytes as having different proteins on their surface. So for example, I know that a CD8 positive T cell has CD8 on its surface. I also know that it has something called CD3. I also know that it has a T cell receptor. I know that my CD4 positive T cell has CD4 and CD3 and T cell receptor. I know that my neutrophil has Ly6G. And you can see this general idea of there are specific proteins on each different cell type. You can think of those proteins as antigens. Just like we have been seeing all sorts of antigen-antibody interactions here. And so, first of all, people have been defining these proteins on the surface of leukocytes since the 70s at least. This is something I had to read during my first week of grad school. The point of me showing you this is to show you that this title is nuts. You don't need to worry anything about the details. And I can imagine that in the past, papers in the past were even worse than this. Because what you can imagine is that here they were looking at a protein called CC chemokine thymus-derived chemotactic agent 4, also known as TCA4, also known as secondary lymphoid tissue chemokine, also known as 6-C-kime, also known as Exodus 2. Like, what? So some immunologists said, okay, this is madness. We need a standardized naming system. We can't just name everything wackily like this. If Henry discovers a protein, and Addie discovers a protein, and they're the same protein, we have to have a way that we can just pick a standard name. We can't call it the Henry protein that's also the Addie protein that, oh, that one time Hannah discovered too, and also Rishi. Like, we have to pick a number, or like pick a standard name for this. Because alternatively, you end up with this business. And so what we did is we gave all of these proteins standardized names. These names freak people out when they learn immunology. They are not hard. They're just standardized names. And the way that they work is that they count. So the first protein somebody found on the surface of a white blood cell was called CD1. And then the second one was called CD2. Yes. We're up in the 300s now. There are situations where like there are A's and B's and like subparts of them, but basically we use this thing called the CD system. Originally we thought that some of these proteins had to do with differentiation, so we called them cluster of differentiation markers, or CD. Now no one cares. It's just CD whatever. It's the name of a protein on the cell of a white, or on the surface of a white blood cell. And so when we want to tell the difference between our white blood cells, we can look for which CD proteins are on their surface. So for example, you can see this T cell had CD8 and CD3. So that's cool, but I can't really see those under a microscope. I can't look at a cell and be like, ah, yes, the CD3 protein. But what I can do is I can use antibodies that bind to those different proteins to help me find those cells of interest. So I can use antibodies that, again, have a modification at their Fc portion that makes them glow. Basically, I use fluorescently labeled antibodies. You can see that I might use an antibody against CD8 with a red tag, and an antibody against CD3 with a blue tag. And then, if I want to find CD8 positive T cells, I look for cells that have both red and blue. I might find other cells that have, say, just blue, because they have just CD3. Those aren't good enough for me. I want just ones that have reds and blues. And so I can just use antibodies. It's the same thing that we've been doing with all these other antigen antibody techniques. It's just now we're doing it on the surface of a cell instead of doing it in sort of liquid form. So does this make sense? I promise you, the CD system, whenever you hear people like mock immunology, they're like, oh, and they talk about CD this and CD that, and I don't know what they're doing, and blah, blah. We're counting. We're just counting. So that's cool, but there's still one other piece to this problem that we have to think about, which is, okay, great, but I still told you I was looking at 10,000 to 1 million samples or cells every time I looked at one of my mice. So how the heck was I looking at 10,000 to 1 million cells and finding the red and the blue ones? Like, you can imagine, yeah, I could do that by immunostaining. But now imagine doing that for 1 million cells. And that would be from each mouse. And at the minimum, I had 12 mice per experiment. How would that go? Anybody want to do that? No, no one wants to do that. Why? Hard. Think about if you had tried to look at 1 million cells per sample in that week last week. So we got around the I can tell the difference between the cells problem. We now know how to tell the difference between all these cells. And we can probably pick out a neutrophil way easier than we could have just by looking at it under a microscope, because we can use the CD markers on the surface of neutrophils. But we still haven't figured out a way to do this and look at a huge number of cells. And so the way that we're going to do that and use a huge number and look at a huge number of cells is using a machine called a flow cytometer. And so the way that the flow cytometer works is we take our sample. That sample is going to have our whole mixture of cells. So this might be the sample that has a whole mixture of cells in it. And we're going to then add some antibodies. So you can see I've got some cells in my mixture that have no antibodies bound to them, because they didn't have the antigens on their surface that I was looking at in this experiment. Some will have just one. Some will have more than one. And so you can see I'm going to have, I'm starting with a heterogeneous mixture of cells that I've labeled with antibodies, and as a result, I'm going to have heterogeneous labeling. I'm then going to take that sample and put it on the machine, and the machine's going to basically suck it up and mix it with a whole bunch of liquid. Which is the sheath fluid. And I'm going to have this mixture, and I'm going to then push it through a nozzle. And when I push it through the nozzle, I'm going to adjust so that I get the cells to actually fall in a stream with 1 cell per droplet of water or per droplet of sheath. So basically, I'm going to kind of use some physics principles to play with water pressure or liquid pressures and things like that, and I'm actually going to make a stream of fluid drops that each have 1 cell per droplet. And I'm going to make this falling array of those cells, so you can see my falling array of cells. Yeah? When my cells are falling, they're going to fall across the path of a laser. So you can see the path of the laser shown here. And there's going to be a detector that is going to detect what comes out of the laser to see how the cells change the laser's light. How do the cells impact the laser's light? There are 3 different ways the cells can impact the laser's light. Someone the other day was asking why I, as an immunologist, had to know physics. Hopefully you know the answer now. So here's my cell. I have 1 kind of cell. I have a 2nd kind of cell. This one at the bottom, it's a granulocyte. See how the granules, they're drawn so nicely? Okay. So if I have the laser coming onto this cell, one thing that can happen is that the cell can actually bend the laser's light based on its size. So the cell can bend the laser's light like that. And if the cell was teeny tiny, there would be a lot less bending. And if the cell was huge, there'd be a lot more. So we have this happening. The detector can detect this, and that will tell it how big my cell is, if I have a big or small cell. This is known as the forward scatter. Which is a measure of size. My laser also is going to go through the cell. If it goes through this cell, nothing really happens. It just sort of goes through the cell. If it goes through this cell, there's lots of stuff in there. And because there's lots of stuff in there, the light gets like bounced around off the stuff. The laser can also the detector can detect that, and that's known as the side scatter, or SSC, which is a measure of the granularity of the cells. Some people also will talk about it as the internal complexity. So we can get size and we can get granularity, just like you got under the microscope, off of this pretty easily. We also will have those antibodies I told you about. We haven't even used the antibody yet. That antibody will have a fluorophore on its Fc, which can react with the laser's light and emit some other light. It glows when the light hits it, and it glows a specific color. So here, we can put on one kind of light, we get another kind of light off. And the detector can detect that light coming off too. It can say what colors were attached to this. So we generally will have multiple detectors. We're going to have some looking at forward and side scatter, and then we're going to have some looking for colors. Was there any red on this cell? Was there any green on this cell? Whatever different colors that we want to look for. The machine that we have here at Drew lets us look for 4 different colors as well as side scatter and forward scatter. So I can look at 6 parameters on every cell. And then when it's all done, the cells go into the trash can. There's a waste tube at the end of the whole thing. When I do this, the machine will move thousands of cells, hundreds to thousands of cells per second. And so, when I was doing those 10,000 to 1,000,000 cells, I was doing that in 2 minutes per sample. By just having each sample run past the laser, getting some information in it about the detector, putting that into the computer. And so for each of my 1,000,000 cells, I would have 6 values. How big, how granular, how much red, how much green, how much of my 2 other colors on every cell. And so then I just get a whole bunch of data that looks like this on my computer. Each dot on this plot is a cell. And this is actual data from my PhD. And so what you can see is I've got my forward scatter, I can plot my side scatter. So I'm basically plotting the size of the cells versus how granular they are. The ones over here are small. The ones over here are big. The ones over here don't have anything inside them. The ones up here have lots of stuff inside them. I can also go ahead and I can even plot that data in different ways. The software lets you kind of look at it like you can look at just the dots, you can look at the contours of the dots to tell you how dense or not dense they are. You can do things like this where you get like colors to tell you about density and you can see the dots. I tell you this because in different primary lit papers you'll see throughout the semester, different people may choose to depict their data in different ways. They all mean the same thing. In addition, so this is some other data. Here we're looking at 2 of the different antibodies. Either do we have an antibody against CD3, which was one color, do we have an antibody against CD8, which is another color, and you can kind of see what the data look like. You can see that I kind of get these 4 clusters of stuff. You can actually also look at this as a histogram. So I can look at how much CD3 and how many cells. And so like you can see I got a bunch of cells that have a lot and a bunch of cells that have a little. Same thing of CD8. I got a bunch of cells with a lot, a bunch of cells with a little. This is actually the same data, just replotted in different ways. So this is kind of cool. But the thing that makes this very powerful is that we have this ability to do something called gating, which is where we can actually use our software to pick out cells of interest and have it tell us about them. So one thing I can do is I can look at this plot where I looked at forward scatter and side scatter of a blood sample, and I can say, you know what? It looks to me like there's 3 different groups of cells. There's this group of cells that are kind of smallish and not super granular. There's this group of cells that are pretty big and medium granular. And there's this group of cells that are kind of medium sized but very granular. So you can see how I could like look at that and say, okay, I think I got 3 groups. I can then use my software to draw little regions around those, which are known as gates, and say, please tell me about all the cells that are in this region. So I could say, I would like to know about all the cells that are in this region. And the first thing it tells me is, well, 60.7% of your cells are in this region. 3.81 are in this region, and 28.4 are in this region. Immunologists also know, whenever they look at blood plots, that actually there always are 3 samples. These are lymphocytes, these are monocytes, these are granulocytes. And so if I look at a flow cytometry of a blood smear or a blood sample, I have 60% lymphocytes, 3% monocytes, 28.4% granulocytes automatically, instead of having to do what you guys had to do in that other lab. I can then also do something else that's really cool. So let's imagine I did this experiment and I said, you know, today I only actually care about lymphocytes. I want to know about the lymphocytes in my mice after I gave them a vaccine. Basically what I do is I double click on the lymphocytes. And I can pull them out and examine them further. And so here I've taken my lymphocytes and said, okay, now I want to only look at lymphocytes. I'm gating on my lymphocytes. And I want to now know in my lymphocytes, how many of them have CD3, how many of them have CD8? I could also say, well, what about my granulocytes? How many of them have CD3 and CD8? What about my monocytes? How many of them have CD3 and CD8? And I can see, oh, I've got 4 different groups again. And I could actually say, I actually only care about this group. Lymphocytes that also have CD3 and CD8. And I can double click on them and keep examining them. And on and on and on. So again, I told you that our cytometer does 4 colors and 2 physical parameters. There are cytometers out there that are doing 40 to 60 parameters. And so you can subset and subset and subset to look at and get percentages and things like that of whatever kind of specialized cell you want. And so you might get something like this where I say, okay, I want to look at just this population of cells. I'm going to look at CD3 versus CD8. I'm going to get this population of just the cells that are positive for both. And then I'm going to look at how much, using my histogram, how much of this other marker they have. Do they have a lot or a little? And so I can sort of analyze the data in that way. Oftentimes, this is really what the data will look like. Oftentimes we draw these boxes called quadrants to help us analyze, although people are doing that less and less. So you can see we kind of have 4 populations here. And they are of different sizes. They have different percentages. If I were going to draw this same plot for you, I would draw circles to be the populations, and they would be big or small based on the percentages. And same thing, if I ask you to draw me flow cytometry plots, you get points for drawing the right axes. So always draw the axes and label the axes first. And then the different populations just have to be big or small circles. And so this is actually a very famous plot that we'll see later in the semester. My class in grad school made this our mascot. The immunology bird, we decided was our mascot because we thought it looked like a bird, because we were kind of being snarky. And that's how the bird looks when you draw it in these fashions. So this is sort of the general idea of flow cytometry. The way that flow cytometry is more frequently used, it will help me with my original question. When I wanted to know how many virus-specific CD8 positive T cells were in my mice at different days, I basically took some blood, ran it through my cytometer with basically an antibody that let me look at virus-specific cells, antibodies that let me look at CD8, found out what percent I had, and then just put that on my graph. So it was all pretty easy. Yep, Sebastian. Because I was working on a project, my experimental question was about how the modification, the thing I was doing to the mice changed CD8 responses. Like that was my project with CD8 cells. So that was why I was looking specifically at CD8 cells. Okay, so we good here. So I have one more slide, which is like one further modification on flow cytometry. But the flow cytometry problem set that I'll post later today, that's not due for a couple weeks, but it's to give you extra info, time for it, basically is involving you reading some data and kind of trying to make sure that you know what's going on in some of these plots, because we will eventually be seeing some of these plots in class. So the last piece of this is a piece that to me as an immunologist is like super important, and I have realized more and more that non-immunologists, like if I talk to Dr. Dunaway, this distinction isn't as obvious to them. So like to me, I'm like, this is a big deal, and they just like lump everything that I've just told you and this next slide into one big thing, and they're like, oh yeah, they're all the same. And I'm like, no, they're not. So this is the kind of flow cytometer we have. It lets us do analysis of our cells, but there is a modification of this, and this modification is known as, fluorescence activated cell sorting, or FACS. Sometimes people use FACS, F-A-C-S, as a word to mean flow cytometry. Because you could say that when you're doing that analysis, you're sorting the cells, right? You're sorting which ones are which. To an immunologist, sorting is a special fancy flow cytometer. And so to me, I only do FACS when I'm sorting, and I'm doing flow cytometry the rest of the time. So if you remember when I described flow cytometry to you before, we had our sample, it went through our machine, it went by the laser, we got stuff in the computer, and then it went in the trash can. Here, we don't go in the trash can. There's a modified part at the end. And so what we have is we actually have two metal plates that the droplet will fall between. We get some data about that cell, and we say either, oh, I love that cell, I like it so much. Or we say, yeah, I don't care about that today. Maybe it's my CD8 cell or not my CD8 cell. Do I care about this cell today or not? I get some information into my computer. I say, I really love this cell, I want it. Or I don't care about this cell. And I set up my computer at the beginning of this. When the droplet falls, it also gets an electrical charge added to it. And then the machine decides, good, bad cell. And the machine actually changes the charges of these plates to make the droplet fall in different directions. And as a result, we can sort the different kinds of cells. And so we could make one kind that just has A and B on it fall in one tube. We can make another kind that has none fall in one tube. And so we can basically physically sort the cells. Having a machine that also does sorting is like 4 times as expensive. And so this will let you get a purified population of whatever kind of cells you want, which can be used for so many cool kinds of experiments. The other piece of this is an immunology fun fact. So you can see that we have this, you can also, there's a video right there of how this works. You can see that we came up with this thing called fluorescence activated cell sorting. And the abbreviation for that was FACS, F-A-C-S. Yeah, makes sense. Very clear abbreviation. So there was also this other person who invented this other machine. This machine called a facsimile machine. You wanted to make fancy copies and send them to your friends, right? That's what you were going to do with your facsimile machine. So let's imagine that you invented the facsimile machine. What do you want to call that facsimile machine for short? You want to call it a fax machine, right? How are you going to spell fax? F-A-C-S. Guess what? The guy who invented the facsimile machine and tried to patent it invented it the year after the fluorescence activated cell sorter. As a result, FACS was already taken. So he had to come up with something else. He worked for a company known as Xerox. And so he got inspired by the name Xerox, and what did he do? He called it a fax machine with an X at the end. So the reason why the fax machine is called a fax machine is because this got discovered first. So that is your fun immunology fact of the day. All right. So now we're going to move into actually doing stuff.